The Earth is 4,600 million years old. Cellular life, according to the latest fossils found, started about 3,500 million years ago. The history of Earth since then is represented in this graph with the main eons and eras. Here you can also see some of the most important events that took place. At the beginning, Earth was not a very hospitable place for sustaining life, with cosmic radiation and a lot of volcanic activity. There was also what is known as the late heavy bombardment, a long period of meteorites falling on Earth that ended about 4,000 million years ago, and it is believed that life began shortly thereafter. Whether the bombardment had anything to do with the beginning of life or triggered in some way the beginning of life is not really known. What we do know is that about 3,500 million years ago we already had some life forms on Earth. They were probably similar to the simplest bacteria we know, Mycoplasma genitalium, which has only 482 genes in a genome of 580 kilobases. Later on, some of these organisms acquired the ability of using sunlight to produce energy, and photosynthesis started. And this, little by little, over a period of millions of years, led to the creation of an atmosphere, increasingly richer in oxygen. This was another very important milestone known as the Great Oxidation Event, which apparently had a lot to do with the diversification of eukaryotic life and with the appearance of multicellular organisms like fungi and plants and animals. Of course, we can study evolution looking at fossils. Scientists have found fossil evidence of cellular life dating back to 3,400 million years ago, and then there are fossils of many of the different species that have populated the planet throughout all this time, like these ammonites that first appeared on Earth some 400 million years ago. Fossils tell us a lot about the morphology and even about the function of these organisms, but of course it would be very useful to have a look also at their genomes, because the genome contains all the instructions required to build an organism. Unfortunately, fossils such as these are made of stone and no DNA can be extracted from them. Therefore, an alternative approach is to look at the genomes of organisms living today. The rationale behind this is that if early organisms were similar to bacteria, their genomes must have been similar to the genomes of present-day bacteria. And if you had early arthropods, for instance, then their genomes must have been similar to the genomes of arthropods, similar to them, because the body plans are similar and the genomes that are incapable of encoding such body plans must have been similar too. So this is basically the issue that we will study in this course, how sequencing the genomes from different species living today will help us to reconstruct the evolutionary history of life on Earth. But first, we must have a look at how whole genomes can be sequenced today. When we extract DNA, we end up with a tube containing a solution with the entire genome of that individual present in many different copies. Traditional sequencing methods were slow, but the process was greatly improved using capillary electrophoresis machines. However, over the last 10 years or so, scientists have developed new sequencing technologies, new machines that can read an entire vertebrate genome in a couple of days for a few thousand euros. One company, in fact, has recently launched a cluster of 10 machines that read 5 human genomes per day at a cost of $1,000 per genome. This and future machines that will be faster and cheaper will enable scientists to increase dramatically the knowledge about the evolution of genomes. Such technologies, of course, are now being applied to sequence the genomes of many different species representing all domains of life, and this is giving us a lot of details about the molecular basis of evolutionary change, something totally unprecedented. Dozens of genomes are now being publicly made publicly available and can be explored using browsers like this one at the University of California in Santa Cruz, where you can activate some tracks that tell you about conservation of the human sequence in, say, 100 different vertebrate genomes. This is the new field of phylogenomics, which is really shedding new light on the mechanisms that explain how life evolved on Earth.